Library approaches to genetic engineering can be either directed evolution strategies or combinatorial methods. In all library methods, there is first diversity generation and then selection of desired variants. Here we illustrate a library of genes in which the one in tan has a property that is distinct from the others. In all cases, we must have a linkage between the gene sequence and the protein it encodes. This can be achieved by compartmentalizing the gene in a cell or micelle. Alternatively, there are phage and ribosome display methods for connecting genotype to phenotype. We then apply some selective pressure to remove all the uninteresting variants and expand the gene variant with the desired activity. There are many ways of generating the genetic diversity, several ways of associating genotype with phenotype, and many ways of enriching the desirable variants. The enrichment process can be either a selection or a screen. In a selection, you apply a procedure to the library population that results in selective survival of the desired population. In a screen, you individually assay each member of the population and choose those with the desired activity. Though directed evolution and combinatorial libraries are genetic engineering techniques, analogs of these processes occur naturally during the production of antibodies in mammalian B cells. Shown here are three crystal structures of FAB fragments of antibodies that each bind a different substrate. I've put them all in the same orientation such that you can see their structural similarity. The blue and yellow regions correspond to the heavy and light chains. Note that we are looking at just the FAB fragment of a larger protein molecule. Mature antibodies are typically IgG antibodies that contain two of these FAB fragments. Thus, an IgG molecule is composed of 4 polypeptides, 2 light chains and 2 heavy chains. Each FAB fragment is composed of just 2 polypeptides, 1 light and 1 heavy, which are in blue and yellow. The CDR loops in orange are the primary source of variability from one antibody to the next, and it is this region of the molecule that binds one specific epitope. Notice that outside this orange region, the molecules are structurally identical. The size of the binding pocket formed by the CDR loops depends entirely on their sequence, which is specific to each antibody. In 1-NSN, we have a very broad, flat pocket. In 2-ADF, we have a more narrow pocket. In 2-DQU, the antigen is a small molecule, and the binding pocket is so small that it is almost entirely embedded within the antibody when it binds. Thus, the CDR loops are highly diverse structurally. Our concern here is how these CDR loop sequences are identified by the immune system. The traditional way to make specific antibodies involves injecting a mouse with the antigen. This process of antibody generation happens within the mouse, and the cells that contain the genes for the antibody and thus produce each antibody are B cells. Different B cells will produce different antibodies. These cells are harvested from the mouse and then immortalized by fusion with myeloma cells. The resulting hybridomas are identified as clones that each produce one specific antibody. Those clones can be expanded by re-injection into a mouse or growth in vitro to produce as much of the monoclonal antibody as desired. So the question is, what is happening within the mouse? In each mouse cell, there are gene clusters that encode the components for constructing heavy and light chains. The heavy chain genes are split into VJ, D, and C coding sequences. There are multiple variable, diversity, and joining segments, that's V, D, and J, and a single constant domain. These VDJ regions encode the CDR loops of the antibody. As a B cell matures, it randomly selects one combination of V, D, and J by recombination of its genome. That VDJ combination specifies a specific antibody sequence that will be expressed by the B cell, and different B cells will choose to produce different VDJ combinations. In this figure, we are showing the maturation of a light chain gene, which is similar to the heavy chain but lacks D segments. All cells in the mouse start with identical heavy and light gene sequences. The heavy chain gene is located on chromosome 14. There are 2 light chain genes called kappa and lambda located on chromosomes 2 and 22. The heavy chain gene has 65 of these V domains, 27 of the Ds, and 6 of the Js. If we calculate how many different ways we can rearrange these domains, there are 10,530 possible ways you can recombine these domains in constructing a heavy chain. Though there are fewer V and J domains in the light chains, These further extend the initial diversity by a few orders of magnitude. Note that this process of VDJ recombination results in a discrete set of potential combinations. It is not an arbitrary population of molecules being generated, it is a large set of specific compositions. The result of VDJ recombination is an IgM antibody. The choice of IgM, IgA, or IgG is defined by the sequence of the C region, and all antibodies start out in the IgM form. The IgM contains 5 Fab regions. The presence of many identical antigen binding regions results in cooperative binding to repetitive structures like virus particles. Thus, a fairly weak binding antibody can nevertheless bind tightly to its antigen, and this is the reason why such a small number of initial diversity is able to bind to pretty much anything. If a B cell becomes activated by binding to an antigen, it will begin the process of affinity maturation. In this diagram, we show the VH domain. VH is the region of the Fab fragment on the heavy chain that contains the CDR loops. It combined with the VL domain are the two balls of protein that directly bind the antigen. Thus, this diagram speaks only of one such domain. The VDJ recombination results in regions of the protein called the hypervariable loops, which is what encode the CDR loop regions of the protein. During affinity maturation, mutations are focused on these hypervariable regions to further improve the avidity and specificity of the antibody. There are several genetic modifications happening during affinity maturation. In somatic hypermutation, a cytidine deaminase mutant mutates C residues to U within these hypervariable regions. This results in GU mismatches, which are repaired in a sloppy manner to generate random mutations. There are additional genetic modifications taking place that result in insertions and deletions of triplets encoding additional codons into these hypervariable regions. This combination of mutations results in amino acid substitutions, insertions, and deletions in the CDR loops. Some of these mutations will improve binding to the antigen, and the encoding B cell will be expanded further than those cells in which the mutation disrupts binding. In parallel, affinity maturation involves class switching. Here, the C segment encoding the constant region of the antibody is switched from one encoding an IgM to one encoding an IgG. With only 2 FAB regions, the IgG must have higher affinity for the substrate than IgM. So class switching increases the stringency of the selection. Through iterative rounds of affinity maturation, the population of B cells make better and better antibodies for their antigens. The VDJ recombination process is analogous to combinatorial methods. It constructs a large sparse matrix of variants encoding many different functions. These processes typically involve large libraries with more than 1 million members, and this large number of variants usually requires the use of selections rather than screens. Combinatorial libraries are most useful for identifying activities that do not already exist, and they typically involve only a single round of diversity generation. The process of affinity maturation is analogous to directed evolution. It involves iterative improvements to a nascent activity through point modifications. In the wet lab, directed evolution libraries are typically smaller, and the enrichment process can be done with screens. They are used to improve or modify a nascent activity through iterative rounds of mutagenesis and screening. Another way to think about this is to consider a topological landscape. The x and y coordinates of this terrain correspond to sequences, and the height of each hill is functionality. Perhaps some of these hills correspond to GFP, RFP, and alkaline phosphatase encoding sequences. Most sequences in this space have no activity, but there are hills where sequences similar in sequence have functionality, with some a little better than the others. In a combinatorial experiment, we start with some region of this sequence space and produce a sparse matrix of constructs with the intent of finding another hill of activity. In this case, the spread of variance hits 4 additional hills of activity, but there are different hills than where our search began. In directed evolution, we similarly start with a sequence on one of these hills of activity, but our goal is to climb to the local maximum activity of that hill through incremental changes.